So what is the single unit that connects you, the giant oak tree in your backyard, and the bacteria currently living on your phone screen? It's kind of a mind-blowing question, right? Because if you strip away all the outward complexity, the leaves, the limbs, the microscopic tails, you're left with this fundamental, awe-inspiring truth. A hidden, microscopic universe is literally the foundation of all life. Welcome to The Explainer. Today, we are unlocking the mystery of the cell. We're going to take a focused, chronological journey starting all the way back with 17th century glass lenses and traveling right up to the incredible mechanics of modern cell biology. Okay, let's dive into this. Here's our roadmap for today. We'll kick things off with the microscopic revolution, move on to the birth of cell theory in the third tenet. Then we'll explore modern cell theory, division and proliferation, and wrap it all up with the power of differentiation. Part 1, the microscopic revolution, revealing the unseen world. For most of human history, our understanding of biology was completely boxed in by what the naked eye could see. It really took a simple technological key to unlock an entirely new biological frontier, the microscope. In 1665, an English scientist named Robert Hooke used a two-lens microscope to look at thin slices of bottle cork. He wrote down what he saw in his famous book, Micrographia. And what he saw was a bunch of tiny, geometrically regular pores. Hooke used this brilliant visual metaphor. He actually coined the term cells, because these little boxes reminded him of the small, unadorned rooms that monks lived in at monasteries. Funny enough, Hooke was actually just looking at the empty, dead walls of plant tissue, completely oblivious to the complex living stuff they used to hold. But soon after, we get this eccentric Dutch dreeper named Antony van Leeuwenhoek, and he takes things to a completely shocking new level. Using a custom single-lens microscope that gave him a massive 270 times magnification, he basically looked at everything he could get his hands on. When he looked at his own dental plaque and later some pepper water, he discovered this completely bustling world of motile, living organisms. He delightfully named them animalcules. And just like that, we had our very first sighting of living bacteria and protozoa. Now, as you look at this timeline, notice something crazy. Compound microscopes appear around 1620. Hooke makes his discovery in 1665, Leeuwenhoek in 1676, and then a massive 200-year gap. The science literally stalled out because the glass lenses had hit a physical limit. It wasn't until the 1850s and 80s that a German engineer named Carl Zeiss, working with Otto Schott and Ernst Abbe, finally upgraded optical lenses to push those magnification boundaries further. Which brings us to part 2, birth of cell theory, drafting the rules of life. So, as microscopes finally improved in the 1830s, scientists started connecting the dots, organizing all these random microscopic observations into actual biological laws. First, you have botanist Matthias Schleiden. He figured out that every structural part of a plant was made of cells, although, side note, he incorrectly thought they formed through a kind of crystallization process from the outside in. But then, Theodor Schwann drops a massive breakthrough. Animal tissues are super fibrous and way harder to study, but Schwann proved that animal structures are also entirely composed of cells. He essentially bridged the gap between the biological kingdoms. He proved that fundamentally, plants and animals share the exact same building blocks. And from the work of Schleiden and Schwann, the first two classical tenets of cell theory were born. Number 1, all living organisms are composed of one or more cells. And number 2, the cell is the most basic unit of life. This was a monumental shift, guys. It dealt a knockout blow to those older vitalist doctrines that assumed life was powered by some mystical, vital principle. Nope, science proved life was built on distinct, observable cellular units. Section 3, the third tenet, and the end of spontaneous generation. Okay, so with those first two rules locked in, one massive, glaring question remained. Where do new cells actually come from? Because for centuries, this ancient Aristotelian myth was just accepted as fact, that life just spontaneously popped into existence from natural heat. While in 1855, German pathologist Rudolf Virchow destroyed that myth with one legendary Latin phrase, omnis cellula e cellula, all cells arise only from pre-existing cells. Though I have to mention the historical drama here. It's widely suggested Virchow actually plagiarized this exact concept from Robert Remak, who had observed cell division through binary fission a few years earlier in 1852. But regardless of who gets the ultimate credit, this final tenet permanently completed classical cell theory. Moving on to part 4, modern cell theory, the upgrade. As the decades rolled on, biology evolved. We moved past just looking at the structure of cells and started uncovering their internal biochemical realities. The classical tenets just weren't enough anymore, so modern biology added a few crucial upgrades. First, Energy flow, meaning metabolism and biochemistry, happens entirely within cells. Second, cells contain the actual genetic blueprints, DNA found in the chromosome and RNA found in the nucleus and cytoplasm. And third, all cells are basically identical in chemical composition across similar species. So the crucial point here is that cells aren't just empty structural bricks. They are incredibly active chemical engines, carrying the actual code of life. Now, what's really interesting about this slide is just the sheer vibrancy of a modern cell. Take a look at this plant cell's complex internal anatomy. We are light years away from Robert Hooke's empty cork walls. Inside that plasma membrane, you've got this bustling factory of organelles: the nucleus, mitochondria, ribosomes, the Golgi apparatus, the endoplasmic reticulum. It's an insanely dynamic environment, perfectly designed to sustain those exact energy flows and genetic blueprints we were just talking about. Part 5. Division and proliferation. Cells in action. Let's transition from the static theories to the dynamic reality of biology. We're going to see exactly how Virchow's third tenet works in real time. Take a look at the colored ring here. This is the cell cycle. A cell doesn't just split randomly whenever it feels like it. It follows a rigorous, phased process. It starts in interphase. You've got the G1 phase where it grows, the S phase where it critically synthesizes and replicates its DNA, and the G2 phase where it rests and prepares. Then it enters that top purple section, the M phase or mitotic phase, which is where the active division takes place. And sometimes it'll even step out into a resting state known as G0. And this brilliantly illustrates the staggering macro result of that cycle. We start with one parent cell up top. Through the exact division process we just broke down, it proliferates. It splits into 2 daughter cells, which then split into 4, then 8, and so on. It creates this cascading exponential family tree of new life, allowing organisms to grow from a single tiny unit into incredibly complex beings made of trillions of cells. Section 6. The power of differentiation. Specialization. So here's the ultimate question. If cells are just rapidly churning out exact copies of one another, how on earth do we eventually get a leaf or a muscle or a functioning brain? Well, that is the final piece of the biological puzzle: differentiation. This is the process where a generic, newly divided cell finally acquires a specific function. To put it simply, it's kind of like a generic office worker finally getting a specialized career assignment. They get tapped on the shoulder and told, hey, go become a specialized leaf cell, or go be a red blood cell. But the biological world is wild, and this isn't always a one-way street. We also have de-differentiation. This happens when a specialized function is actually removed from a differentiated cell. It's this amazing biological phenomenon where cells can basically quit their jobs and revert right back to a generic, unspecialized state. And wrapping up the terminology, we have redifferentiation. This is when that recently de-differentiated generic cell acquires a brand new function. It can be completely reprogrammed. And this highly dynamic ability to assign, remove, and reassign cell functions is exactly the fundamental concept that makes things like advanced plant tissue culture, cloning, and modern biotechnology even possible today. Which brings us to the end of our explainer. We've journeyed from Hooke's empty cork walls to the incredibly complex, reprogrammable chemical engines that literally sustain our existence today. Understanding cell theory is absolutely crucial because it's the fundamental rulebook for medicine, for agriculture, and for our own lives. But I want to leave you with one profound, unsolved evolutionary mystery to spark your curiosity. If Virchow's third tenet holds true, and every single living cell strictly arises from a pre-existing cell, where exactly did the very first cell come from? Think about that for a while. Keep learning, and I'll catch you on the next explainer.